Hello, I'm Nomi Harris, and it's my pleasure to welcome you to the first OBF BOSC ISCB COSI webinar. So what exactly is the OBF? The Open Bioinformatics Foundation is a nonprofit, volunteer-run organization that promotes open source software and open science in the broader biological research community. Some of our major activities are coordinating the annual BOSC conference, participating in the Google Summer of Code, and serving as an umbrella organization for member projects. The first BOSC was in 2000. The OBF was actually founded the next year, growing out of volunteer projects such as BioPearl, BioPython, and it was incorporated that year as a nonprofit. Bylaws were enacted in 2005, and in 2012, we became an associate project of the Software in the Public Interest organization. This is the current board members. We will have 2 new board candidates standing for election at our upcoming public board meeting, which actually is 1 week from today, Tuesday, September 21st, 17 o'clock UTC, and you can read more about that on our blog. We have 2 candidates running for board member at large and 2 things that we are going to have a membership vote on. One is proposing a new grant program based on the OBF event fellowships that is aimed at supporting grassroots projects that are running events in their own communities. We're calling it the OBF Community Support Sponsorship. And we are also introducing a code of conduct that will apply to all of OBF. We've already had one for BOSC, but this will be for the broader parent organization. So I mentioned the event fellowships. This started in 2016 as a travel fellowship. And then when travel became unwise, we changed it to be an event fellowship. It's aimed at increasing diverse participation at events that are related to open source bioinformatics and open science. We have given over 30 awards so far for people to attend BOSC and other conferences. Most of our awardees have written blog posts about their experiences. You can find that on our website. Currently, we have 2 application deadlines per year. There's one coming up on October 1st, and you can find out more and apply on our website. We hope you'll spread the word to people who might be interested. The OBF is open to anyone who is involved in or wants to become involved in promoting open source or open science in the biosciences. And it's easy and free to apply for membership. You can find that on our website. And in addition to our website, there are other ways to contact us. You can email us. We have an open Slack, and we have an organization on GitHub. And there is an account for OBF called OBF News on Twitter, and there's also a BOSC account on Twitter. So that's OBF. Now I'll tell you a little more about BOSC, which I've been chairing for the last 10 years. So BOSC, as I mentioned, has been held every year since 2000. So we just had our 22nd annual BOSC, numbering from 0. Usually it's been part of ISMB. We cover all aspects of open star bioinformatics, including open source, open science, open data, and more. So we were part of ISMB ECCB in July 2021. In 2018 and 2020, we held a joint event with the Galaxy Community Conference. And this year, as we have for the past few years, we followed BOSC with a free and open collaborative work event called CoFest. This was the BOSC 2021 organizing committee. We also had a great group of reviewers who reviewed all the submitted abstracts. We had 3 keynotes at BOSC. They were all really great. You can find more on our website and see the videos on our YouTube channel. I wanted to mention some of the things that we've been doing to promote accessibility and inclusion at BOSC. This year, we offered free registration actually to 20 people from around the world. You can see the map there of their locations. This included a special round of the OBF Event Fellowship that was made possible by our sponsors. We also enabled those who were submitting abstracts to just check a box on the abstract submission form if they felt they needed assistance with the ISMB registration fee. This year was also the first year we offered honoraria to keynote speakers, with the recognition that not all researchers are equally privileged to be able to gift their time. And keynote speakers who didn't want to or were not able to accept an honorarium were able to donate it back to the OBF to support future inclusion efforts. This year was also the first time we had a keynote talk that was not in English. We had a speaker from Cameroon who gave his talk in French, and we had English subtitles added to his talk. And as I mentioned, we have a YouTube channel where you can find all of the BOSC talks. We made them available immediately after the conference, and they have closed captioning that you can turn on. So as with OBF, we have multiple channels to communicate with us. These links are in the footer of every BOSC webpage. You can follow us on Twitter, look for our YouTube videos, and join us on Slack. I hope you'll join our community and join us at BOSC 2022. So that's the end of my introduction about OBF and BOSC. And now I am pleased to introduce Dr. Mad Price Ball, who is the Executive Director and President of the Open Humans Foundation. They're also affiliate faculty at the Center for Research and Interdisciplinarity in the Peer-Produced Research Lab. Mad's focus is on enabling people to access and use their health and personal data. They also gave the keynote talk at BOSC 2017, and were one of the panelists for our panel on open data standards, opportunities, and challenges. So Matt is going to be telling us about open sourcing ourselves together. Over to you, Matt.
Picking out the screen here. Great. So I'm excited to share some of my work and lessons learned, experiences in a sort of broad array of ways in which we might think about how open applies to understanding ourselves as humans. I mean that in the biomedical sense, in understanding our health data or genomic data, but also in a broader sense, to call to the desire and empowerment of people to understand themselves as an intrinsic desire, as part of being human, as individuals and communities. Any story starts somewhere, and I will start mine with how I became enchanted by the magic of open. I was a graduate student doing molecular biology and omics research, and I had to give lab meeting. And for lab meeting, I needed image of DNA, the molecular structure of it. So like any good grad student, I went to the internet and I landed on Wikipedia, and I found my image. This is what I found. And I put it in my slide, and then reviewing my slides, luckily before I actually presented this, I realized it was actually embarrassingly incorrect. The hydrogen bonds made no sense. Oxygen doesn't bond to oxygen, and there were missing oxygens on the ribose rings. And so a little bit horrified, I took that away. And of course, I had to make my own image now. I needed one. And so I went ahead and did that and put it on Wikipedia. to save others from the potential mistake that I had stumbled into. And from there, I had learned how to contribute into that system. And I went on to add new pictures and diagrams, mostly with the genetics theme, because that's what I knew. I edited articles, in particular the genetics article, bringing it to a featured article status. And it was rewarding to be able to produce things that I knew were something that others were going to see, learn from, and use. I also had fun. I added pictures of cats. There are good reasons for them. That's temperature-sensitive mutation, the Siamese cat. But part of the fun here is also that there's a little inside humor. There's actually advice, internal and inside joke, that you should not be adding pictures of cats to Wikipedia. And this is to note that part of the appeal, especially as I continued with this, was not merely the act itself, but the community and culture and social interactions that exist within the people, the Wikipedians in this case, that are engaged in creating things together. A couple years later, my skills became relevant in a way that was more powerful. I happened to be in the right place at the right time with the right skills to help put a version of Wikipedia on one laptop or child laptops. And this was offline. Many of these laptops went out to children in Peru, and so hundreds of thousands of children were able to get access to Wikipedia due to this effort. It was ad hoc, unplanned, and I would never have anticipated it. And a powerful outcome, in my opinion, for something that actually didn't, in retrospect, take that much time. It was luck, but it was also possible because of open things. Not merely that the content of Wikipedia was open, but that there was open source code that people had shared that was adapted to compress files and images to create this. And not just that, but also that One Laptop per Child as an organization had a strong open ethos, and its governance was also open, in this sense that a volunteer initiated effort that didn't have a top-down designation could actually be welcomed and included and make it out onto the laptops, consuming a significant amount of storage space on them, but with a significant reward. So I was doing all that, and it was a little unrelated to the molecular biology research I was doing at the time, but these things then came together because the place I was working had another project which seems to combine these themes. So what does open mean when researching humans? The project I started working with, and then for many years was really leading a lot of the coordination and management of operations, was the Personal Genome Project. I was in George Church's lab at Harvard Medical School. The Personal Genome Project is one of George's many big ideas. And in this case, it was one that a lot of people were inspired by. And it had a big ask. The big idea, which was radical, was to take extensive personal data, genomes, and then extensive phenotype, health data, and in essence, everything else. It's because geneticists are very sort of self-centered, and they think that everything else is phenotype. But to take all that, whatever people were willing to share with phenotype, in addition to the genomes, and make full public data release. It's a sort of literal sense of saying we're going to open source ourselves. And it was bold in part because this data is considered dangerous or privacy issues. And so it's a concern. And so some of the lessons I take from this going forward is understanding how to go ahead and work with doing open in a context where we know there's risks to people, for example, risks with privacy. So the issues of scary data are not unique to genome data. They're not even unique to health data. I think that in the years since the Personal Genome Project started, we've become more and more aware of all the other data that's out there with big data and Facebook and Google and Twitter and whatnot out there in the cloud collecting our data. So the conversations are there and more laws have been passed. But how can we be empowered with it? Well, some of the lessons in genome data are worth reflecting on because it contains properties that point to exactly why this data is both of interest and worrisome. Genome data contains potential health information about an individual. It might tell you about the risks of diseases and those diseases might not be treatable. Alzheimer's risk is an example where a relatively common genetic variant might greatly increase one's risk, and there's not much to be done about it. And so the medical utility is questionable because it's not actionable if there's nothing to be done. And yet the privacy concern seems fairly relevant. One can imagine if you're in your 50s or 60s, you might wonder if people would be a little less inclined to invite you to lead long-term positions of management if they suspect that your mental state is going to deteriorate. At the same time, genomes contain ancestry information, inescapably so. This can mean distant ancestry like ethnicity and race and surname prediction, or also, and of huge interest to most direct-to-consumer purchasers of this data who go and seek it out is relatives, is your nearby relatives, finding your cousins, your third cousins. And so ancestry DNA has really taken off, and there's a lot of this data out there now in the form of genotyping. And it's also interesting to reflect that many of the adverse events that have occurred in this data weren't about health. There was a lot of concern around enabling people to discover something like their Alzheimer's risk. But in my observation, a lot of the unexpected and distressing and dramatic outcomes have been about unexpected half siblings or children or surprise cousins. And I've seen it in someone close I know. These are real outcomes and surprisingly common. What this means is that genomes are combining two different aspects of data, and they can't be separated. One is that they are meaningful. They contain information that matters to you, and it matters to you if others know. And they are identifiable. If something was identifiable, but the information in it was trivial and not particularly meaningful, then it wouldn't be a concern for others to find out. Conversely, if it were meaningful but not identifiable, then it's less of a concern that others might find out that it's yours. It's that combination within the data that makes the data particularly scary. And it's not just the genomes that became a concern in research. Cells have genomes too. And that's something that the Personal Genome Project was built around, and it created cell lines, human cell lines, that were making an effort to be more ethical about their creation, recognizing that the material within those cells was also genetic data. Other types of data have these same properties, identifiable and meaningful. This is location data. This is my location in New York City when I lived there a couple years. And you can see that I was mostly in one of two places, that is home or work. And this pair alone makes location data highly identifiable, even when rendered fuzzy and reduced to zip code level of specificity. There is still a remarkable amount of uniqueness in location data. And at the same time, we all know that location data might be meaningful in a way that people would really prefer to keep private. And so genome data has served as a place where the lessons learned are also lessons relevant more broadly around personal data. So as part of this process, I also had the joy of engaging participants who were enthusiastic about participating and contributing themselves, donating their data to a greater good as part of this project. And it really was a matter of meeting them, in part because sample collections had to occur, but also engaging them in a continuous way, because informed consent is not just a one-off experience, especially in this case, but to answer questions and help them understand what it is they're participating in and the data that they're choosing to make public as best they can. Because the project was asking people to do something unusual, to take on this potentially large unknown risk of making that data public without potential benefit. Usually a risk in human subjects research is considered acceptable because the individual stands to potentially benefit. And here the benefit was not one that one would normally consider in the risk-benefit analysis of altruism, of a goal for advancing mankind rather than the individual. But it went forward, and I think that is remarkable. It was a very engaged, informed process. So there was a quiz to help check understanding and to help individuals understand the data. One of the things I did was help create reports that would match the genome against existing public knowledge of genetic variants, not to try to warn them or notify them of any potential medical information. But the ethical principle here was that when they go ahead with taking on this risk of sharing the data, to try to do our best to empower them to understand what that data means as a researcher would understand that data. Risks are hard to predict and understand. Any analysis of a genome is going to be subject to our understanding of what data means at the time, and risks may be unknowable. It can make the conversation difficult because one might pose risks that are ultimately highly unrealistic and completely miss the ones that are actually going to occur. And yet, we are consenting to risks in daily life. And so what's different? Why do we worry about protecting people in this process of open sourcing their data for science? We actually share data with lots of big tech companies without such an extensive process of concern. One of the aspects here is that there is a balance of power between researcher and subject. A researcher has access to knowledge and understanding and can try their best to convey this to the subject, but it's never going to be complete. It may be far from complete. It's very hard to do this. And there's been a lot of research into like trying this with videos, with quizzes. And the reality is doing so is hard, in which case we offload a lot of this oversight to a committee to decide, like, this is too much an ask, this is too big an ask. We can't expect people to understand how big it is. I think it's interesting insights because it means that, like, our method of trying to manage this with big data and consent and expecting individuals to consent is ultimately just as limited, because we've tried our best in science to do this, and it's hard. But there is an interesting counterpoint, and that is self-research. Our understanding of oversight has been largely focused on this dyadic divide between the researcher and the subject. But what does it mean when the individual is the researcher? I have a quote here from Hans Jonas, an old essay, which set aside attempting utilitarian analysis and instead thought about sort of the moral rights of individuals. And he pointed to the researcher as being this important starting point, as the single most appropriate person to consent to research, because the researcher knows, understands at best the risks and potential benefits, and is also driven by that goal. The motivation rests within the researcher to engage in discovery. Jonas also suggested that from there one might expand research subject recruitment to within researchers themselves as being the individuals who shared the ideals and understanding of what research entails. And self-research has an important role in scientific discovery. One of the most well-known examples being H. pylori and ulcers, and Barry Marshall wittingly drinking the bacterium to test and demonstrate the causality of this bacteria causing the condition. And yet it's rare. It's rare for self-research to really make an impact. Part of what I find interesting when thinking about self-research or self-experimentation is that in a world where we have data and software, and where we do research using those tools, and less about we're trying to do it with chemistry and wet lab equipment, it means in theory, it should be more possible to do individual self-research and innovate in that way. And so it's a place, an opportunity for open tools that can be shared much more easily, because there's no physical objects. And so that marginal cost should be negligible. So what power do we have to do that? What power do we have to research ourselves as individuals and communities? So my work with the Personal Genome Project inspired a new project called Open Humans. And Open Humans took that experience with data to expand beyond genome data, and to center this on the individual, to enable bringing data in, diverse personal data into a personal account, and then opt in to share it with various activities and projects. So an academic researcher could create a project that invited donation of data to create new research and potentially reuse that data. Open Humans did not limit itself to a dyadic divide. My work with the Personal Genome Project, and knowing how much people wanted to understand their genomes and do science and not just contribute was that I wanted to enable people to do this themselves. It's once they have that data, should they not be able to engage in research themselves? And so community projects also exist, and it means that communities have created citizen science projects and data commons that give them a more democratic ability to engage in research. There's also support with Jupyter Hub and Jupyter Notebooks for individuals to create and share the ability to analyze their individual data in private. And so as a researcher, you may want to start by doing it yourself privately, and the code for that analysis becomes a shareable object. So now we've moved on from the principle of sharing the data as being the good that enables others or advances knowledge to sharing the code for performing an analysis such that others can use it and run it and see the same analysis of their own data. So this is a snapshot and it's a couple of years old, but it represents that there are a variety of data sources and sharing, and activities could do one, the other, or both. They might be a tool that adds data, tool that invites a data sharing and contribution to a project, or they do both. And the vision here was that by doing this within a platform where other activities could reuse components, it meant that that we had more of a reusability of these tools. So a data import tool or action engaged by an individual for one project could enable another project that is using the same data. It feeds in and reinforces itself as in its network effect, one hopes. All that said, in all of those efforts, it was very difficult to get data reuse to be the amazing good that we might have hoped it would be. And this isn't to say it was not useful, but data might actually be harder to reuse than we might appreciate. I think that because it has this absence of negligible marginal cost, it's tempting to think that data is magic, to be overly optimistic. But it's actually harder to work with, and this might be especially appreciated by anybody who's done so, who has done bioinformatics. One of my favorite analogies, and I hope my attribution to Matt is correct here, but this was in comments just made after talks, people talking to each other at this symposium hosted by Quantified Self. And it was that data's not fluid. It's like luggage. You have to go to a lot of work to figure out what to pack, and then you have to move it, and then you have to unpack it, and then you realize that you forgot to pack something. And I think a lot of us have been there. That's for an initial use. Data can be even harder to reuse because it's designed to fit original needs. And so this is the stuff that got packed might not contain the information that you need to answer your questions. You needed something more, something else, and it just wasn't there. In addition, if we rely, and I think this is important and we should do this, if we rely on individual opt-in and understanding and informed consent to sharing data, that means that there's not going to be a lot of people that are going to be involved in going to get their data to look at it themselves or to contribute it. And so this really narrows the amount of reuse that can occur. And yet, I think that working with those individuals, that very phenomenon where we are concerned that it requires all that individual engagement, is also an opportunity, because if you have that individual motivation and engagement, you can do new things with data reuse that make new things possible. So it might not have contained what you needed, but you can do something that original data collector didn't do. You can connect it to disparate data that they didn't have. The individual can go and collect 2 different data sources. They can choose to collect new data. They can start tracking something new because they know that there's a project that would combine these. They can also choose to try an intervention and see the effect on that data. So working with individuals is powerful. And who is motivated to do that, to engage? Why would they care? In many cases, solidarity and altruism is powerful to help their community. This is often a patient community that is concerned with improving care and understanding about their particular health condition. It can also be intrinsic curiosity. People just want to understand more about the data or themselves and explore it and see what's out there. And that's an important a value to have as human beings and support. They may have a need, personal health issue that they want to solve, and they are frustrated with the medical establishment. These all connect to self-research. In the end, some of these aspects connect in a way that makes, in principle, this type of research more possible. The individual has access to their own data or potentially to access to choose to collect new data. They also are a person that is more likely to have motivation to use their personal data in new ways. And I think this potential of self-research connects us back to open in a broader sense to understand how anyone can do things with their data and engage in discovery about themselves. And this can be something that may feel entirely unrelated to health, but in other important and powerful cases, it really does relate to health. And people have used self-research methods to address and greatly improve chronic, complex conditions. Innovation often starts with individuals. So the more people that can do this, the more potential innovation might be possible. And enabling more people to be able to do this means that we empower individuals and communities to do more for themselves. And so an inspiring example of this is Dana Lewis and the Open Artificial Pancreas System. Dana has type 1 diabetes. And before any commercial device was available, Dana built off of some existing open source software and connected the dots to use continuous glucose monitor data to automate a basal insulin pump and connect the loop. That's why it's called an artificial pancreas, such that she wouldn't have to be manually adjusting the amount of insulin that was coming in. And now there's commercial devices doing this, but this was one of the earliest starting points or sparks in what expanded to a much larger community of loopers, of type 1 diabetes patients that were already very tech and data savvy because they constantly monitored this data, engaging and doing more for themselves and empowered to share their tools and understanding and also interventions. But it's hard. But it's hard. If we go back to self-research and we think, okay, this is an opportunity, there's access to motivation, it fumbles on ability. Using data is hard. Actually, doing science is hard. I wish I could tell you, okay, how about we just create an app to analyze the data and that's going to solve everyone's problems. But I think it's actually more complex than that. There's no app that's going to solve everything or address all issues because this is this becomes a question of doing science. Figuring out the right question to ask, finding the right tool, designing protocols and implementing them, analyzing data and interpreting it. It feels like it should be a simple staged process, but for anybody who's actually done science, you know that each one of these steps turns into an arrow that takes you back to an earlier step. Troubleshooting is pervasive. A protocol might be too hard to do, and so you have to change the protocol. You might discover after analysis that the data is garbage and you need to go back to the beginning and change your tool. You might discover after you've put it all together that your interpretation is ambiguous and you need to go back and redesign the protocol or change the question. Solving these issues is hard, and even better to anticipate them so that you don't conduct the entire experiment only to find ambiguous interpretation. Takes a lot of experience. It is a type of knowledge that is not easily implemented as a tool or a textbook or a paper. The phrase tacit knowledge was coined by Michael Polanyi decades ago to describe a reality of science, that scientists don't learn to do science by reading papers. They learn it by doing it together with other scientists, with teamwork, with meeting each other at conferences, with lunchtime chats, and with lab meeting. Like me as a grad student, putting together those slides. That was learning how to do science, was talking and sharing my ideas and getting feedback. And Polanyi knew what he was talking about because while he was engaging in philosophy or sociology as he looked at this issue, he was already a well-established researcher and had made important scientific discoveries and advances. So he's quite the polymath. So how do we do that? How do we enable self-research? How do we enable more people to engage in science when they're not going to be attending graduate school for self-research? The Keating Memorial Project is a project we started in early 2020, and we wanted to know how we could make the process of doing self-research more of a collaborative endeavor where people could learn from each other and use things others had learned. And it quickly evolved into self-research chats, which I'm going to say looked pretty much like lab meeting. It enabled people to share ideas and get feedback and encouragement and form social connections. So they knew who to go to ask a question of. We've looked into it with interviews. And so here we are moving into sociology, and we can see that these aspects of sharing with others do provide value to individuals where they can learn from feedback and they're motivated to share what they've learned with others to benefit others in addition to themselves. So that brings me back to another picture of Open Humans, where Open Humans isn't a technology platform, it's a community. And there's these weekly and regular meetings via video chats, and as well as online chat in Slack and an online forum. And discussion spans issues around self-research and citizen science projects, as well as tech development and ideas, which might be around how to get data or use data or analyze data, and as well as the platform itself and the tools it might have. So I've become super interested in self-research, but when I think about self-research, I don't mean to limit it to an individual. This collaborative aspect is important, and it's been largely absent, perhaps, because self-research is such a self-contained process. But self-research, in a little more metaphorical, broader sense, might be considered how communities of people can collaboratively co-create projects by themselves, or how individuals in communities might lead a project that others in that community contribute to, that represents their interests. And so I wanted to close with two projects that highlight this. One is Transbiome. Transbiome it might be seen as a way a community has been empowered to study itself. It is initiated by a trans woman with a question that is specific and relevant to transgender people, to trans women, and that is to understand the microbiome of the neovagina. This is an organ that has a real health need for individuals that have it, and there is very little research in even characterizing what is there. It can be subject to potential infections and such. Clara is a student, and this project, while the idea came from a community member and the motivation to do so came from that, has two other members of the team, Bastian, my colleague at CRE, and myself. Clara is a student in Bastian's lab. And we brought in expert experience that Clara wasn't going to have. That is the tacit knowledge. For example, just in this last week, we've had data from a sequencing service returned to us, and we can see issues in it. And there's all sorts of questions about what to do about that. Is it our fault or are theirs? How do we communicate concerns? What should we do? These are situations that are pervasive in actual lived science, and situations where a graduate student is likely to need a more senior experienced individual to help navigate it. Earlier in the project, navigating the whole process of ethics board approval was something that I had experience with, and so it was not so scary to me. And I can only imagine for citizen scientists trying to bootstrap something themselves, like getting through some of these aspects of doing research is very difficult, and a high amount of tacit knowledge about what a typical solution is, or what to anticipate and avoid is just necessary, and in many ways impossible to render explicit until doing it together with others. Another project I wanted to share was Quantified Flu. Quantified Flu came together during one of our Open Humans community calls, and it really did take advantage of Open Humans infrastructure to pull together data from various sources. The idea, and this is not unique in the height of the pandemic as it was getting started, was wondering if we could use this activity tracker data to predict if we were getting sick, to detect it. Unlike academic research in this topic, this project was really co-created and interested in giving an individual insight. And so it was logging data and then creating visualizations. So an individual could see how their heart rate had changed recently, and how symptom tracking had occurred over time, and how these data correlated or changed over time. They could see their own changes. It had a significant amount, a very substantial contribution in terms of code and adding new data sources like Garmin and Apple Watch, things that might be of use later. And it had sustained use, which was unusual for this mobile health field. It was characterized by continuous development and contribution. And so it was iterative. And that's another way in which open processes might be difficult to translate to much of our standard academic research practice, because when we do human subjects research, we try to have an event-based review up front and design everything up front and then approve it. But here, what it was a changing over time as new contributions came in and evolved and responding to ideas. But the co-creation process meant that it was a high fit to user interest and had a high engagement. And it was just published on September 10th. That's just a few days ago in JMIR. And the main finding of this is related to understanding how it was developed as a co-created project. It's not making a claim to an insight and being able to predict infection, but rather how this process created something that had such a high use rate and was successful. So with that, I wanted to say thank you. That should be Gary Wolf, not Gara Wolf. To the many people that I have the joy of interacting with, Bastian in particular and his lab at the CRE have been a major collaboration for me in the past couple years. And also a lot of thank you to Gary Wolf and Quantified Self, and also to Stephen Jonas, who's just like, as an individual, has lots of great ideas. Gary and the Quantified Self community have been great to learn from. And then so many people, there's too many to list, that were in self-research chats and beyond. And I want to close with encouragement to anyone who has stuck it out and listened to all of this, is to say that I'm going to guess that you're somebody who does science and has skills. To encourage you to not be afraid to leave your lane, because you have knowledge that is useful in other domains, in research and data and analysis. And also to recognize that open is also a social process. It's beyond simply sharing the artifacts and applying the appropriate licenses. And while it does often start with a lead individual sharing what one is doing in a social sense and not merely those artifacts, as something that enables the serendipity of collaboration. So with that, I think we'll go to questions.
Thanks so much, Matt. That was great. I saw a question appear and then disappear. I'm not sure if here we go. How do you deal with data sovereignty issues between family members in an open project?
So there's interesting ethical questions, and I think that this question may be explicitly wondering about genetic data. So if I share my genome, how have I impacted my children who share half of it and don't have any capacity to make a decision on that? How do I impact my siblings or parents who also share that genetic material? It's not unique to genomes. We have all sorts of things that we do share that involve other people. We share photos we take at parties. We share other aspects of our life and stories from our life that include individuals. And so I think in one sense, we do need to recognize that individuals have autonomy in choosing what they share, and that they have a power to share things that harm others. And what's important is to educate them to understand that there may be people in the background of their data that are also being shared. And so when they're making that decision, to be aware of that and mindful of asking permission if appropriate, or making decisions around that.
Thank you. So I have a question regarding sort of how you frame data reuse. So a lot of people like to talk about the FAIR principles: findability, accessibility, interoperability, and reuse. And I may have missed it, but I didn't hear you explicitly mention that. But I realize that that formulation is controversial. And so I wondered what your feelings were about FAIR.
So part of it is that when I've worked with data reuse, it's been, I would say, like, the FAIR principles have had a very internal sort of researcher to researcher facing conversation. And so I don't tend to think in those terms because I'm thinking about access, control, ability to reuse for the people that the data came from. And it's rare for individuals to refer to FAIR principles in that. I'm not sure I have like much more to say about that. I do think that data is functionally an artifact where it's easy to sort of overestimate in principle its utility, its actual use in the future, simply because something is potentially usable doesn't make it so. And there's got to be more than simply releasing data to realize the potential benefits in it.
Yeah, that makes sense. So is there anything that you're doing to try to encourage reusers to keep the metadata attached to the data sets?
Metadata is itself like, what is metadata, right? And like, these conversations are great. Matt Beats had I mentioned him in the talk, and he studies the process of research as a sociologist. And he's like, one person's metadata is another person's data. And so, like, what is metadata in addition to what is appropriate metadata is so contextual to the original design of the data creator. So it's going to be designed to meet, just like the data itself, whatever you call within that data to designate as metadata is going to be designed to meet the needs of that original data collector and user. I don't worry too much about trying to enforce sort of a universal metadata standard. It's nice to know like when a data set was created or whether it was modified, where it came from. But yeah, when we talk about this Wild West of everything from your Twitter posts to your genome data, it can be hard to have any universal standards.